Hello, my name is Kara Peak, and I'm a doctoral student in Dr. Victor Varner's lab. And today I'll be presenting my research on determining the effects of FGF10 in the embryonic lung. So during development, the embryonic lung airway undergoes a series of branching events to build the bronchial tree that is collectively known as branching morphogenesis. A key molecule necessary for proper airway branching is fibroblast growth factor 10, or FGF10. It is thought that focal regions of FGF10 expression pattern the lung mesenchyme, the tissue surrounding the airway, and specify the location of new branches, possibly through localized proliferation. However, physical mechanisms such as fluid pressure and cellular contractility have been shown to modulate branching as well. It still remains unclear how FGF10 elicits the physical and cellular mechanisms that drive airway branching, and I therefore aim to answer this unknown fundamental question within the field by using FGF10 soaked beads as ectopic sources of growth factor to recapitulate the hypothesized focal patterning and attribute any changes along the normally unbranched ventral epithelium to these sources. Now, when we culture these lungs in the presence of the focal FGF10, we surprisingly see the formation of not one, but multiple supernumerary buds, which exhibited a characteristic wavelength. Because FGF10 is thought to initiate buds via localized proliferation, we investigated its role in supernumerary bud formation and found by inhibiting proliferation using acetylcholine, reduced the number of supernumerary buds. Next, to directly quantify rates of proliferation, we used EDU incorporation and saw focal sources of FGF10 significantly increase the number of EDU positive nuclei within the ventral epithelium. It therefore seems that the formation of supernumerary buds relied on elevated epithelial proliferation. Now, because the supernumerary buds relied on proliferation and formed multiple buds with a characteristic wavelength, we were reminded of some previous work using mesenchyme-free explants that were able to undergo branching via a growth-dependent buckling mechanism, wherein the multiple branches formed with a characteristic wavelength and simultaneously. We therefore wanted to investigate the dynamics of our supernumerary buds using time-lapse microscopy and saw the multiple supernumerary buds indeed form simultaneously. We therefore hypothesized a growth-dependent buckling mechanism was driving the formation of the multiple supernumerary buds. To further validate our buckling mechanism hypothesis, we next modulated epithelial tension via luminal fluid pressure to prevent or enhance epithelial buckling. First, using an activator of epithelial secretion, forskolin, luminal fluid pressure was increased and cast the epithelium into tension, thus preventing any buckling and supernumerary bud formation. Now, going the opposite direction, we use an inhibitor of epithelial fluid secretion, bumetanide, to decrease luminal fluid pressure and cast the epithelium into compression earlier. Indeed, we saw the decreased fluid pressure cause supernumerary buds to form significantly earlier and with a greater number and smaller wavelength. Altogether, I have shown a focal source of FGF10 is able to cause the ventral epithelium to grow, and surrounded by the constraining mesenchyme, it buckles to form multiple supernumerary buds. The formation of these buds can be modulated by epithelial tension through luminal fluid pressure. These results demonstrate FGF10 is capable of eliciting a physical buckling within the intact lung, and the hypothesized focal patterning of singular FGF10 source to new epithelial branch is too simplistic of a model, and we must consider physical mechanisms when thinking about branching morphogenesis. We want to further expand this work by creating a biophysical model that will allow us to test the effects of mechanical perturbations on branching. Additionally, we want to experimentally test the effect of other mechanical perturbations, including exogenous fluid pressure and tissue stiffness. Altogether, these data will further answer how both cellular and physical mechanisms are responsible for branching morphogenesis, and not only answer this largely unknown question, but provide insight into techniques for tissue engineering applications. Thank you.